Our topic is artificial insemination in cattle and buffalo. Artificial insemination. Artificial insemination is the technique in which semen with living sperms is collected from the male and introduced into the female reproductive tract at proper time with the help of instruments. Collection of semen. Selection of breeding bulls. Bulls selected should be healthier and particularly born to high yielding mother. Preparation of young bulls. Quality bulls calves should be procured at 6 to 9 months, and at 12 months the bull should be screened to check whether it's free from disease or not. Bulls selected must be finally screened to detect any chromosomal abnormality. At 18 months of age, bulls should be trained for semen collection. At 24 months of age, semen will be collected and evaluated before the final selection. Once the bull is selected, maintenance rations should be provided. Collection yard. Semen collection yard should be near to the laboratory. It must be clean, not windy, must be closed surface. Ground must be roughly covered with concrete. If semen is collected in ethaned flow, then it should be sprinkled with water to avoid dust in air. Teaser animal should be tied within a service crate. If the dummy is used, it should not be taller than the bull. Cleanness of bull. The bull which semen is to be collected should be clean and free from dirt to avoid contamination of semen. Underline of bull is wiped with towel before semen collection. If it's too dirty, wash it referred but done too long before collection process. Hairs in the underline has to be cutted to 4 to 7 centimeter length. Selection of teaser cow. As far as possible, cow should be a same breed, color and size. It must be able to bear weight of bull. Better to select a cattle which gone through 2 to 3 calvings. It better to tie its tail at one side. Dummy cows also used vastly. Collection methods of semen. There are vastly two different methods are used. They are artificial vagina method and electrostimulation method. Artificial vagina method. The artificial vagina has the following parts: a heavy hard rubber, two loaves open at both ends with a nozzle for air and water in and outlet. Inner sleeve of rubber or rubber liner, the semen receiving cone or rubber cone. Semen collection tube made of glass or plastic. This is the diagram of longitudinal section of an artificial vagina process. Before using the semen collection, all the parts are washed thoroughly and sterilized properly. The rubber liner is inserted into the hose. The water jacket of the artificial vagina is filled with hot water at a temperature of 45 degrees Celsius. By opening the nozzle, semen collection tube is fixed to the narrow end of the artificial vagina hose. The artificial vagina is lubricated with sterile jelly to a length of 3 to 4 inches. Air is blown through the nozzle into the water jacket to create pressure in it. AV collection process. The cow or dummy is secured in the cervix crate. When the bull mounts the cow, the operator directs the gland penis into the artificial vagina. Then the bull ejaculated in the artificial vagina. Air and water nozzles are opened to release the pressure in the water in it. Then the semen is collected in the collection tube. Electrostimulation method. It is the process of obtaining semen by passing a small value of electric current into the rectum of the animal. Procedure: rectum of the bull cleaned with 6% of NaCl solution. Then the electric probe is inserted into the rectum for about 12 inches. Then the electric current is passed at 5 volts. Then the return to 5 volt to 0. Then after 10 to 15 seconds, 5 volt is increased. Then that come to 0. The cycle is carried out until it reaches 15 volt and the sperm collected. This is the picture of electrostimulator. Semen evaluation should be evaluated closely for abnormal appearance. The presence of the small clots or blood can indicate such conditions as seminal vesiculates, color, volume, consistency, count, specific gravity, abnormalities, and so on. Parameters: normal values. Ejaculate volume 5 ml, range 1 to 15 ml. Sperm concentration 1200 million per ml, range 300 to 2500 million per ml. Total sperm per ejaculate typically 4 to 5 billion. Progressive motility greater than 30%. Morphology greater than 70% normal. Storage. In 1949, British scientists discovered that the addition of glycerol to the semen extender improved resistance of sperm to freezing. There are two methods of freezing and storing semen: dry ice and alcohol, liquid nitrogen methods. Liquid nitrogen is preferred because there is no evidence of fertility deterioration with the age. Frozen semen can be stored indefinitely if proper temperature is maintained. Fresh liquid semen can be successfully stored for 1 to 4 days at 40 degrees. Semen is usually stored in liquid nitrogen tanks. Artificial coloring is frequently added to semen. Frozen semen is packed in single doses of glass vials or plastic straws at 5 degrees Celsius. The final level of glycerol should be 7.0 to 7.6 percentage during the freezing process act. Antibiotics are added to inhibit bacteria and to kill pathogenic organisms. Semen to be diluted in such a way that 1 ml of extended semen will contain 20 million alive sperm cells. Liquid nitrogen plays a vital role for storing the frozen semen straws at a temperature of -9 to 196 degrees Celsius for longer periods. This is the picture of liquid nitrogen container and the semen straws. Insemination. The inseminator will get ready by wearing a plastic apron, gumboots, and gloves. The semen straw, after thawing, is loaded in a sterilized AA gun. The inseminator will insert the gloved left hand into the rectum by applying a soft soap or other lubricant on the glove and back rack the animal, and the hand is further inserted and will catch and hold the cervix through the rectal wall, and insemination with the artificial insemination gun. This is the picture of the person inseminating a cow by holding the cervix. These are the advantages of the artificial insemination. There is no need to maintenance of breeding. It prevents the spreading of certain diseases. Better breeding efficiency is ensured by high possibilities of getting pregnant. The semen collected can be taken into any places. It is helpful to inseminate the animal and refuse to stand or accept the meal. Cost efficient, reduces physical damage. Disadvantages of artificial insemination: requires well-trained operators and special equipments, requires more time process than natural services. Improper cleaning of instruments and the sanitary conditions may lead to lower fertility. If the bull is not properly tested, the spread of genital diseases will be increased. Thank you.